So hello students and welcome back again. In today's video tutorial we would be talking about C elegans. इस आज के हम वीडियो ट्यूटोरियल में C elegans के बेसिक फंक्शंस के बारे में पढ़ेंगे. What is this C elegans? What is the function of C elegans? What are the properties of C elegans and why this C elegans is being used as a model organism? Basically the questions that are asked from C elegans are from the introductory section that we all would be dealing as well as from the vulva development. So as you can find on your screen, you can find the C elegans. The C elegans is just 8 to 10 millimeter long nematode. It belongs to the class Chromodorera, the order Raptidia, family Raptidae, and the genus is Caenorhabditis elegans. Caenorhabditis elegans have been known for the formation of a vulva. What is the vulva? You can just look at your screen. आप अपनी screen पे देख सकते हैं that there is an opening which is present in the case of female C. elegans. This opening is responsible for laying the eggs into the external environment. From this opening, the eggs are being laid into the external environment. This opening is referred to as the vulva. C. elegans have been known for the discovery of the cell apoptosis pathway. The cell death pathway has been discovered in the case of C. elegans, as well as are one of the most important animals in the case of developmental biology. सेल डेथ पाथवे जिसे हम एपोप्टोसिस बोलते हैं वो सी एलिगेंस में ही डिस्कवर्ड है. डेवलपमेंटल बायोलॉजी में वुल्वा का डेवलपमेंट जिसने आगे चल के रिप्रोडक्टिव ऑर्गन्स को बनाया ह्यूमंस के अंदर, उसका भी डेवलपमेंट जो है वो हम सी एलिगेंस के अंदर सबसे पहले स्टडी करते हैं. Why C. elegans is being studied? What is so specific about C. elegans? That is why this is chosen as a model organism. First, they are free living. These organisms are completely transparent. So as they are inside, they can be visualized from outside without dissecting them. क्योंकि ये ऑर्गेनिज्म ट्रांसपेरेंट है तो इसीलिए आप इनके अंदर देख सकते हैं कि किस तरीके के स्ट्रक्चरल चेंजेस इनकी बॉडी के अंदर हो रहे हैं विदाउट क्लीविंग देम. They are about 1 millimeter in length, lives in a temperate soily environment. Their body is non-segmented. Okay, so their body is not segmented into a large number of segments as in the case of our earthworm. These are vermiform and are bilaterally symmetrical as that of humans. जैसे हम bilaterally symmetrical हैं, so they are also bilaterally symmetrical. Has cuticular integuments, four main epidermal cord, and fluid-filled pseudo-coelomate cavity. They have a fluid-filled cavity which is present in their body. You can just recognize C. elegans as an as a miniature version of Ascaris lumbricoides. जैसे आपने Ascaris lumbricoides के डायग्राम को देखा था, याद करिए एस्केरेसिस जो कॉज करता था, मेडिकल में इसके बारे में काफी इंफॉर्मेशन थी. तो एस्केरिस लुम्ब्रिकॉइडिस के एक मिनिएचर वर्जन को ही आप सी एलिगेंस की तरह कंसीडर कर सकते हैं. बट द सी एलिगेंस इज ट्रांसपेरेंट, इट हैज हाई रिप्रोडक्टिव रेट and it generally feed upon a colony of bacteria. So these can be effectively maintained within a petri plate as they are going to feed upon the bacterial cells. So they can be effectively maintained on a single petri plate. C. elegans are hermaphrodite organisms. That means they have both male as well as female reproductive organs present within the same organism and can be even males. Okay. Next. Males have a single lobed gonad, vast difference, and has a tail specialized for mating. So they had a tail through which they perform a specific function in the case of mating. Hermaphrodites have two ovaries, oviduct, spermatheca, and a single uterus. Hermaphrodite gonads produce first sperm and then oocytes. So, firstly, these hermaphrodite gonads which are present, firstly, they have a temporal speciation. That means first they produce the sperms and then they produce this oocyte. ये जो organism है ये hermaphrodite तो है ही, लेकिन पहले sperm produce करता है, बाद में egg produce करता है और वही स्पर्म एग को फर्टिलाइज भी कर सकते हैं and can lead to the formation of an offspring. So why we are studying this organism? See, you can look at the miniature version of this particular organism as well as the transverse section. In the transverse section, you can find that we would be moving from the anterior till the posterior part. In the anterior part, there is the pharynx which is present. So it is going to respire from this anterior position. There is lumen of intestine. Now there is the cavity which is present. This cavity is commonly referred to as the vulva. And this cavity will be the most important cavity that we would be studying. ये जो vulva है, ये ही सबसे important cavity है जिसे हम लोग study करने वाले हैं यहाँ पर. It is this vulva that we are going to talk about a lot. The development of this vulva involved 22 different cells. 22 cells will be involved. And these 22 cells will form a circular cavity around the vulva. It is this vulva only from where these embryo, the developing embryo, can move outside the body. Okay, so it would be this vulva that will serve as the site from where the developing embryo can be laid outside the body. But when the vulva will not be produced, जब vulva का development नहीं होगा because of mutations associated, supposingly mutations takes place. If mutations occurs in these cells forming the vulva, then what will happen? The embryo will not be made, will not be able to move outside. As the embryo will not be able to move outside, so there would be a patch, there would be a group of vulval cell, there would be a group of embryo being entrapped within the body of the female. फीमेल की बॉडी के अंदर ही बहुत सारे एम्ब्रायो क्या हो जाएंगे, इंटैक्ट रह जाएंगे, so that we can have genetic study on all the embryo that would be produced. And the most important questions in CSR have been asked from how this vulva development takes place. CSR के अंदर सबसे ज्यादा क्वेश्चन vulva development से पूछे जाते हैं कि किस प्रकार से vulva development हुआ है Caenorhabditis elegans के अंदर, and which are the genes that are going to take part in the case of vulva development. Okay, then you can find that there is the spermatheca. The sperms are there where the development of sperm is going to take place. The germ cells that are leading to the formation of the oocytes. Ultimately, the oocyte and the spermatheca are going to fertilize with each other, leading to the formation of embryo. Spermatheca से sperm oocyte के साथ में combine हो जाएंगे. When the spermatheca sperm combines with the oocyte, they form embryo. जब sperm fertilize कर देता है oocyte को तो वो embryo बना देता है. Embryo body के अंदर uterus में ही packed रहते हैं because this is the uterus. Then through the vulva they began to move outside. Okay, and if this is being inhibited, then embryo patch will be produced within the body of the female that we would be studying. Next slide. Just a second. This is, as you can find, the organism which is hermaphrodite is referred to as XX, whereas the organism that is referred to as male is written as XO. Okay, so these are the two different types of sexes which are present in the case of Caenorhabditis elegans. C elegans के अंदर ये दो तरीके के sexes हैं, जैसे कि आप यहाँ पर देख रहे हैं. hermaphrodite organism में vulva है और vulva के अंदर ही यहाँ पर eggs हैं जो uterus के अंदर हैं. Then the sperms will be there and then the embryos will be produced. Whereas in the case of male, there is no vulva, there is no egg, there is no uterus which is found. Okay, so the Caenorhabditis elegans can remain either in the form of a male or in the form of a hermaphrodite. जेनेटिकली अगर इसे जेनेटिक इसका मेकअप देखते हैं तो जेनेटिक जैसा आपने देखा एक्स एक्स हो सकता है या एक्स जीरो हो सकता है. एक्स जीरो इज रेफर टू एज मेल, एक्स एक्स इज रेफर टू एज एन hermaphrodite organism. Now you can find here predominantly C. elegans is the hermaphrodite producing both sperm and eggs, thus can self-fertilize. Predominantly, if you are going to talk about C elegans, what are C elegans? यहाँ से सुनिए, बहुत ध्यान से सुनिएगा. C elegans होते क्या हैं? C elegans are basically hermaphrodite organisms. They are they are usually hermaphrodite. Sometimes they can be male. कभी कभी मेल भी हो सकते हैं, लेकिन अधिकतर टाइम पे वो hermaphrodite ही होते हैं. उनमें मेल रिप्रोडक्टिव ऑर्गन्स भी होते हैं और फीमेल रिप्रोडक्टिव ऑर्गन्स भी होते हैं. They have about 10 eggs laid inside. The older eggs are laid about as fast as new eggs are being made. Okay, so as soon as the new eggs are being made, the older eggs are being laid outside after the act of fertilization in order to form the new zygote. जैसे जैसे न्यू एग्स का फॉर्मेशन होता है, ओल्ड एग्स को रिलीज कर दिया जाता है, and they fertilized with the sperm, leading to the formation of zygote. Oocyte passes into the spermatheca. So see, it is this oocyte that is going to pass into the spermatheca. You can find just before the vulva. आप देख सकते हैं vulva से पहले यहाँ पर eggs हैं और vulva के बाद में ventral surface of the body की तरफ sperms हैं. So what you can find is that oocyte passes into the spermatheca and are fertilized. Oocyte spermatheca की तरफ बढ़ते हैं और यूट्रस में फर्टिलाइजेशन हो जाता है. It is these oocytes that moves towards the spermatheca. Within the uterus, the process of fertilization takes place. And now the embryonic development is going to be continued. Embryo develops in the uterus and have a few cleavage before the egg are laid. So the embryo has a few dozen cells when the embryo are laid. The embryo develops into worm about the next 8 to 18 hours. Now the embryo is being produced after the act of fertilization has taken place. And once the act of fertilization has taken place, few dozens of cells have been produced. As the few dozens of cells have been produced, now the embryo will be made to move outside the vulva and will be laid in the external environment. अब जब एक बार एम्ब्रायो बन जाता है तो एम्ब्रायो को वुल्वा से बाहर निकाल दिया जाता है और बाहर जाके एक्सटर्नल एनवायरनमेंट में एम्ब्रायो का डेवलपमेंट होता है. Next, advantages of C. elegans as a model organism. Very important. Why C. elegans has been selected as a model organism? C. elegans have been selected as a model organism because of several features. They have simple body organization. They have the simplest, one of the simplest body organization is there, as well as they are completely transparent. They can be easily grown over the bacterial plates. Okay, so you can just take a bacterial colony, bacterial plate, or a bacterial lawn over which all these C. elegans can effectively grow. This is the bacterial lawn that you can take, and over the bacterial lawn the C. elegans can effectively grow. Okay, here I'm just taking a bacterial lawn. Supposingly if this is a bacterial lawn, then C. elegans can effectively grow over it. They have a shorter lifespan. They have a very smaller lifespan. As they have a very smaller lifespan, so they can produce very large number of springs. So they have a shorter lifespan, just of 3 days, but could produce very large number of offspring. They have powerful genetics because almost all the genes of the C. elegans have been understood. Their genetic interaction has been understood, that how does the genes interact with each other. तो इसीलिए क्योंकि हमें आज जीन्स का इंटरेक्शन पता है कि जीन्स इंटरेक्ट कैसे करते हैं, तो आज हमें ये भी पता है कि जीन्स का इंटरेक्शन कैसे होता है, जीन्स का डेवलपमेंट कैसे होता है, जीन्स का ट्रांसमिशन कैसे होता है एक जनरेशन से दूसरी जनरेशन के लिए. सो दे आर यूज्ड एज अ पावरफुल मॉडल ऑर्गेनिज्म in the case of genetics. Their body is completely transparent. As their body is completely transparent, so the inner development of the internal development of the body can be effectively visualized. They have a comparatively smaller size. Okay, they are invariant. They are fully described. Autonomy as well as development could be fully described, as well as the development of the first RNA interference technology is also based upon C. elegans, so that the C. elegans can be effectively modified. तो हम C. elegans को मॉडिफाई कर सकते हैं. And as we can see, modify the C elegans, so different types of features can be introduced or different types of effects of genes can be understood. तो आप temporal knockouts बना सकते हैं. So first, if RNA interference is being used, we can make two different types of knockouts. One is referred to as the temporal knockouts. What do you mean by temporal knockouts? Temporal knockout or permanent knockout means temporal knockout means knocking out a gene for a particular period of time. Knocking out a gene for a particular period of time. For a particular period of time. And its effects could be understood. As well as the permanent knockouts could be produced. Another type of knockouts is permanent knockout. Permanent knockout means you can permanently inactivate certain genes. Permanently inactivating genes. Inactivating genes. Okay, so that could be done. Next point. Now, these are the simplest organisms with a simple nervous system. They are all eukaryotic, share the same cellular and molecular structure as is being shared by the eukaryotic cell. So they grow, these organisms go through a complex developmental process, including embryogenesis, morphogenesis, and growth into an adult organism. So they undergo simple type of the developmental processes, including embryogenesis. They will lead to the formation of embryo. They will lead to morphogenesis in order to form adult organism, and will result into the growth of a adult organism. Next, the biological information obtained from C. elegans may be directly applicable to more complex organisms because C. elegans are simple organisms. So the biological information that we are going to derive from C. elegans can be used in the case of complex organisms. देखिए जो बायोलॉजिकल इंफॉर्मेशन हम सी एलिगेंस से ड्राइव करते हैं उसे हम कॉम्प्लेक्स ऑर्गेनिज्म में डायरेक्टली यूज करते हैं. अबाउट 35% ऑफ द सी एलिगेंस genome shows exact homology with the human genome. 35% सीक्वेंसेस की एग्जैक्ट होमोलॉजी है जो ह्यूमन जीनोम के साथ में प्रेजेंट है. These are some data that you have to remember. I mean, I am coming to the main point. C. elegans was the first multicellular organism to have its genome sequenced. Young fleshy starved larva best survives freezing in 15% glycerol solution, can be stored at minus 80 degrees Celsius for a very longer period of time. In such type of period of time, they form a Dwar modification. Now, what is this Dwar modification that we are going to visualize? देखिए लार्वा को हम बहुत ही कम टेम्परेचर पे स्टोर कर सकते हैं सी एलिगेंस के, दैट इज approximately minus 80 degrees Celsius over a time period of 10 years. So you can store them at minus 80 degrees Celsius for a time period of 10 years. That makes this organism very useful for research. Okay. About 35 to 45% of the worm stored in liquid nitrogen are going to survive for an enormous longer period of time. बहुत ज्यादा समय के लिए भी आप इसे स्टोर करके रख सकते हैं. और जब इसे स्टोर किया जाता है तो एक मॉडिफाइड वर्जन ऑफ लार्वा बना लेता है. That is referred to as the Dwar modification. Chromosomal and genetics, I have already told you that it has six number of chromosomes. The number of chromosomes in the case of model organism, number of chromosomes for model organism can be effectively remembered by the trick. And what is the trick? The trick is PDACP. PDACP. These are all model organisms. This P stands for Saccharomyces pombi. ये जो मॉडल ऑर्गेनिज्म है उनका एक ट्रिक है जिसके आप थ्रू आप सारे मॉडल ऑर्गेनिज्म के क्रोमोसोम्स नंबर को याद कर सकते हैं. Saccharomyces pombi is a budding yeast and it is haploid. It has three number of chromosomes. Drosophila melanogaster. Drosophila melanogaster, which is commonly referred to as the queen of genetics, is referred to as four number of chromosomes. Okay, this is haploid. This is A refers to Arabidopsis thaliana. We have just finished this plant and it has five number of chromosomes. Okay, it has five number of chromosomes. C refers to Caenorhabditis elegans, and the Caenorhabditis elegans has six number of chromosomes, whereas P, last P, refers to Pisum sativum. This Pisum sativum has also seven number of chromosomes. So 3, 4, 5, 6, 7. PDACP means Saccharomyces pombe, Drosophila melanogaster, Adopsis thaliana, Caenorhabditis elegans, and Pisum sativum. So, my dear students, we have finished Pisum sativum in the case of Mendelian genetics. We have also seen just before these lectures began about Arabidopsis thaliana. Now, we would be looking at Caenorhabditis elegans, and then the Caenorhabditis elegans will be followed by the Drosophila melanogaster. Okay, next, these organisms are hermaphrodite. They have two X chromosomes, so they are can be written as XX, whereas males can be written as X0, having one chromosome instead of a pair of hemizygotes. So instead of a pair of two chromosomes, so males are said to be hemizygous in nature. देखिए मेल्स के अंदर दो क्रोमोसोम नहीं है, मेल्स के अंदर केवल एक ही क्रोमोसोम है, इसलिए मेल्स को हेमिजाइगस भी हम कह सकते हैं. नेक्स्ट, so life stage, as I was telling you, that there is a very specific life stage in which the Caenorhabditis elegans can survive the harsh environmental condition. It is referred to as DOR. DOR is referred to as harsh environmental condition surviving state. Harsh environmental condition, harsh environmental, environmental condition surviving state, condition surviving state. Okay, this is the state that is present. This is referred to as Dwar modification. Life cycle. It lives a total number of 2 weeks. The eggs are fertilized within the adult hermaphrodite and laid in about the 40 cell stage. Eggs को लेके जब किया जाता है, जब 40 cell stage हो जाती है, they hatch and it produces approximately four larval stages. It is going to produce four different types of larval stages, and those are referred to as L1, L2, L3, and L4 larva. L3 larval stage is commonly referred to as the dwarf modification. They hatch and it proceed through four larval stages. C. elegans can adopt an alternative life cycle called as the dwarf stage in unfavorable condition. देखिए clearly लिखा है, ये जो ड्वार स्टेज होता है, ये एक लार्वा की एक स्पेशलाइज्ड स्टेज है जिसे हम L3 स्टेज कह रहे हैं. इट इज बीइंग फॉर्म्ड इन द अनफेवरेबल एनवायरमेंटल कंडीशन. ये बिल्कुल देखिए बैक्टीरिया जैसे स्पोर्स फॉर्म कर लेते था ना, वैसे ही ये यहाँ पर ड्वार फॉर्म कर देता है. क्योंकि ये डोर फॉर्म कर लेता है, इसीलिए हार्श एनवायरमेंटल कंडीशंस को बाईपास कर पाता है. अब ये डोर फॉर्मेशन के लिए जो जीन्स होते हैं वो कौन से होते हैं? देयर आर होम्योटिक जीन्स दैट आर इन्वॉल्व इन द फॉर्मेशन ऑफ डोर लार्वल स्टेजेस. homeotic homeotic genes are involved, and it is only because of the dwarf formation. It is only because of the dwarf formation the Caenorhabditis elegans can be stored in liquid nitrogen for a very long period of time. Dwarf larvae are thin and can move out, but their mouth are plugged and they cannot eat and can remain viable for 3 months. They can remain viable for 3 months and under certain conditions for years. Dwarf larva appears to be non-aging. देखिए ये बड़ा इंपॉर्टेंट है. डुआर लार्वा के अंदर एजिंग होती ही नहीं है. ये अपने क्रोमोसोम्स के एंड्स को मेंटेन कर लेता है. Okay, they have the very unique ability to express telomerase enzyme and to maintain its chromosomal end. जब इसके क्रोमोसोम्स के एंड मेंटेन रहेंगे तो ऐसा लगता है कि इसके अंदर कभी एजिंग होती ही नहीं है ड्वार स्टेज के अंदर. सो ड्वार लार्वा appears to be non aging. They can remain for months एंड देन री एंटर इनटू द L4 स्टेज। ये L3 स्टेज में कई महीनों तक, कई सालों तक भी रह सकते हैं, और फिर अगली लाइफ स्टेज में आ जाएंगे L4 में, when they encounter the suitable amount of food resources and the good environmental condition. Under harsh environmental condition they form door. जब भाई हार्श एनवायरमेंटल कंडीशन हो जाएगी तब तो डोर बना देगा, और जब favorable environmental condition आ जाएगी, then the life cycle is going to be continued. Next, so study with C elegans. Now, what is the contribution of this small nematodal organism in the case of genetics and molecular biology? It has some very vast number of contributions. बहुत ज्यादा contribution है C elegans का. सबसे पहले अगर हम बात करें RNA इंटरफ्रेंस की जो शुरुआत है, RNA इंटरफ्रेंस की जो डिस्कवरी है, वही C elegans के अंदर कराई गई थी. It was only in the case of C elegans that RNA interference has been discovered. C elegans can be modified very effectively by feeding them over transgenic bacteria. You can feed the C. elegans with transgenic bacteria or injected with RNA into the body cavity. Then they are going to express the double stranded RNA component of the gene, thereby causing RNA silencing. It can lead to RNA silencing. It can cause RNA silencing to take place. Certain RNA can be made to silence. Next, live samples of Caenorhabditis elegans used to explore the effect of zero gravity on muscle development and physiology. So, live samples of Caenorhabditis elegans have been taken to the International Space Center, where they have been used to study muscle development and physiology. By live जो सैंपल्स हैं सी एलिगेंस के, उनको हम यूज करते हैं जीरो ग्रेविटी में मसल के डेवलपमेंट को पढ़ने के लिए और फिजियोलॉजी को स्टडी करने के लिए, दैट व्हाट इज द फिजियोलॉजी of muscle development and how does the muscle development takes place. The research was primarily about genetic basis of muscle atrophy, which relate to space travels. Okay, next. So this is the basic about C. elegans. Studying meiosis is considerably simplified in the sperm and egg nucleus of the gonads. The difficulty of heterogeneous cellular population are eliminated because every nucleus at a given position in the gonad therefore is at roughly the same step in meiosis. Because in the case of mutational studies, अगर हम vulval cells के अंदर mutation कर दें, तो जो गोनाड्स का फर्टिलाइजेशन होगा वो एक साथ में होगा, वो एक साथ में डेवलप करेंगे, and they are roughly at the same stage of meiosis, so we can get fundamentally correct results as compared to other organs. क्योंकि जब आप मॉडिफाई कर देंगे वुल्वा को तो वुल्वा ओपन नहीं हो पाएगा, तो बहुत सारे एम्ब्रायो किसमें रह जाएंगे, फीमेल की बॉडी में ही रह जाएंगे. दे विल फॉर्म एन एम्ब्रायनिक पैच and every gonad will be present at the same stage in that embryonic patch. embryonic patch बन जाएगा फीमेल की बॉडी में. हर एक गोनाड जो है वो सेम स्टेज पे आ जाएगा जब वो फीमेल की बॉडी में प्रेजेंट होगा. Next, it can also be used to study nicotine dependence, etc., and other different types of disorders have been studied. Just a second. Okay, screen sharing stopped. Why? Just a second. Okay, so here we were. So it has been used in order to study about nicotine withdrawal syndrome, tolerance, sensitivity, etc. So this is all about the key important features of the Caenorhabditis elegans and how the Caenorhabditis elegans has been used as a model technological tool for exploring the